How many are non-CS? 3. अच्छा, those many. Okay. ठीक है. What's your name between Yash? Okay. Yatharth. है ना? ठीक है. Okay. All right. So this is just fun, है ना? We'll finish in 20 minutes, 30 minutes. More is about get a flavor of how you would go through a research paper. That is what the goal is. So it's probabilistic sample spectral cluster. I am taking this because there was extra time, है ना? So that is why I'm taking it. So what's the motivation? So motivation from this is coming that agricultural productivity is not even in India, not just in India, but other around the world as well. It's affected by excessive heat, scarcity of water. In fact, when this project started, we had a French collaborator who said this Arctic blasts happen, no, in Europe and US where a lot of snow comes and then the crops get damaged. So you have to now, with the change in climate, you have to take care of this resistance as well. So you have to somehow develop species which are, resistant to these kind of traits. So there, what we do is if you can cluster these plants and into certain groups, then you can tell, okay, this was the gene. Let's say you grow one plant and you come to know that this is, resistant to some insect. Then you can cluster them and you can say, okay, maybe this set of plants are resistant to some diseases. That's where they're close to each other. So clustering them well is an important task for the plant biologist, and it keeps going on because they keep deriving new species, keep finding new species. है ना? So that is what it is. So you need to find efficient clustering, highly diverse species, and you will then breed them. You will find, take one or two from this group which are resistant to disease, take one which is resistant to snow, take one which is resistant to drought, and try to, mutate it or breed it and make a plant which is resistant to all of them. So that is what the goal is. And clustering sometimes takes a lot of time. So underline you want to do some sampling as well. So we'll talk about that. This I have told you many times in the class that every living organism has an associated whole genome sequence with it, WG, which you have done problem also now, right? And what is a single nucleotide polymorphism, or SNP, is where these genomes, whole genomes start differing. If you look at it, GCA, they differ at the third point, and we pick this and we call it as SNP or SNP, and this is something very important for the plant biologist. So that is what we work with rather than working with the whole genome sometimes. An easier way to work with them is working with phenotypic data, which phenotypic is a fancy name of saying physical data. So if you look at this table, first column is just going species, and then you've got some characteristics. Early plant vigor, which is poor, good, very good, EPV. Then pH is plant height. NPB is number of primary branches. NPPP is number of pods per plant, and so on and so forth. So these numbers are given to you. Now you can do a clustering based on genetic data, you can do clustering based on phenotypic data, you can relate genetic data, phenotypic data, you can find the proteins inside them. There's a lot of work which can be done in this bioinformatics domain. Today what I'm telling you is only looking at the phenotypic data. There is another paper which we have published on genetic data, but today the focus is on phenotypic. Okay. So how do people usually use? Now, did anybody go through the clustering PDF I had uploaded on the drive? Have you gone through different types of clustering yet? Anybody has looked at it? Okay. Let's take a look a little bit. You don't even do you are the sincere lot who comes, you don't do the problems also. I came to know you had not done all the practice problems also. Never mind. So this is supporting material, and this is the clustering survey, and the paper which I am going over today in the slides is this one, real life paper. So this is Pierre J, and when it was published in 21, it was the most downloaded paper in the plant science domain. It didn't lead to that many citations, which I thought would, but at least a lot of people looked at it because nobody had seen it from that angle. So the reason I was referring to this clustering survey is there's a good set of table here. maybe it's towards the end. It was a horizontal table if I remember it correctly. Look. So you should kind of spend time on this a little bit in case even survey paper you have is doing clustering, or your research has any component of clustering, you should take a look at this. Hai na? So if you look at it, look, there are different categories based on partition, based on hierarchy, based on fuzzy theory, distribution density, graph theory, grid, fractal, model, kernel, ensemble, in swarm, quantum, spectral. So the one which I'm referring to today is going to be this spectral clustering, which we are using in this work. We'll tell you why, because it's highly accurate. That's what it says. The accuracy is high, although it's expensive as well. So there are many such techniques, but hierarchical clustering is one which these people have used in the plant domain. Okay, going back to the slide. Okay, let's come back. So I told you the motivation, genetic phenotypic data, and we were here. So hierarchical clustering is that standard algorithm these days. So out of those lists, you can see there was one based on hierarchy. So that's the one they use. Although it suffers from high computational complexity and low accuracy issues. So the cost of it is high, and it's not considered to be one of the most accurate clustering either. So we said what we'll do is we'll apply spectral clustering, which I showed you in the table. It is considered as one of the most accurate. What does spectral mean? What is spectrum or a spectral? Frequency. From the mathematical, this is more electrical. From the math point of view, what did it be? What is the spectrum of a matrix? Yes, Yadhat. The set of eigenvalues, right? Eigenvalues together is called the spectrum in the math domain. In electrical, you may refer to as frequency. In fact, eigenvalues sometimes map to frequencies as well. So that is why frequency of a particular object or of a device which you're making. Okay. So spectral clustering is considered to be highly accurate, but it's expensive as well. It's order n cube. N cube would be really bad. If n is a million, imagine how much it will be 10 to the 18, it'll become that much. So it will be really expensive. So we don't want to do that. So we say we're going to use it cheaply. We're going to do sampling as well. So so the novelty of this work was that nobody had thought of applying this spectral clustering and sampling to this plant genetic data. And applying it is less than straightforward. That is what I want to show you in a real life. So machine learning has many components, right? The machine learning which you have learned with me is model based, right? Which means that tomorrow you can go and devise your own model. For example, we hear generative AI, right? We hear LLMs and large language models and all that stuff. So people decided that although we didn't cover neural network in this class, which was part of computational intelligence, convolution neural networks, deep neural networks, a kind of neural network becomes a transformer, and then you add an attention mechanism and do so. And so you derive new models. So that would be fundamental core AI which you would do, which we call. The one which I'm talking here is applicable AI, where I'm taking the existing algorithms and applying to a domain area. And it's less than trivial because application itself requires some decision making, which we're going to talk about today. So that's the goal. So these are just few slides on why you would want pivotal sampling, although my focus is less on sampling, more on clustering today. So you can just think of sampling as picking certain species out of the book and trying to cluster them. Once we have done them, we take the remaining species and assign them to the clusters. How you're doing sampling, just forget about it because that's not what the goal. The only reason we picked pivotal was because it was linear. The complexity had to be low. We used that. So this is the algorithm for pivotal sampling. I'm again going to skip it because that's not I want you to learn today in the class. But what is important in this is when you sample anything, you have to assign probabilities to every unit in a typical most of the sampling. That this is the probability of this unit, this unit, this unit. And then the units get decided which one gets picked and which one doesn't. So you can see in this slide, I've written that you obtain incursion probabilities of all the units. And then you do a selection step and a rejection step, which you can ignore. The important part is every unit, when you're doing sampling, it has to be assigned some sort of a probability. है ना? Okay, so now you can see the slide changes to green. So this is where our important contribution came. We want to assign a probability to every gene which has a certain set of phenotypic characteristics. Remember early plant vigor, NPP, NPP. Every species has that. We want to assign a probability to it so that either it gets selected or it gets rejected, so that we have a smaller sample. Instead of 1,000,000 data points which we had species, we may end up with only 100,000 or 10,000, and we want to do that. So to assign the species, what we did, the first equation which you look at, we normalized it. This is common in machine learning, that the range of a particular phenotype characteristic could be from 1 to 100,000 or 10 to the 23. You don't want this variation to create numerical errors, so you normalize it, which is the first step. The second step is we find something called as a deviation. Now, what is a deviation? We say that, okay, early plant vigor, high value is good. Number of pods per plant, high value is good. So higher the value of these characteristics, the better it is for us. So we're going to assign deviation saying find the maximum value of a particular characteristic and tell how much am I as a species away from it. So this is what I'm doing. I'm taking the maximum value of a characteristic and saying how far I am. So if you had to look up at the table, let's say what you'll do, you'll take plant height. Higher is better. So the maximum value in this is, let's say 67. So I'll say how much am I far from 67? Let's say if I'm the first species, I'll say 67 minus 4. This is the deviation I have for this one characteristic. But I have to do it for all the phenotypic characteristics, which means the next one I will take, okay, I am the highest, so the deviation is 0 plus. Then the next I'll say I am not the highest, this is highest, 68 minus 68. This is the deviation I am away from the maximum value. Now, and I'll do it for SWL, for DPI, and I'll add all the deviations for individual characteristics. Why I am adding these deviations? Because what I know that if I am closer to the maximum value, I am good. So I'll give it a higher value if the deviation is low. So I'll have an inverse relationship. So 1 over deviation becomes my inclusion probability. So if you have a low probability, then get it picked. Low deviation, get myself selected in the group. That's what I'm trying to tell. So that is what we have said. Yeah. Highest value is? Correct. So that's good. It's good you're thinking, Anup, right? So what Anup is saying, that what if the maximum value which you picked turns out to be the noise? Then what should we do? Any idea? You tell the solution. I was coming to this. Let me finish this thought and then we'll say. So like I said, we said deviation, low deviation is better. So inclusion probability is inverse of that. This is another normalization, so you can ignore it, 1 over summation. This thing you can just ignore. We're saying inclusion probability is inversely proportional to the deviation. But now coming to Anup's question, why are we finding the deviation from the maximum value? Is it representing, what do we want? Which, let's say I've got 10 balls. I want to pick those balls, let's say are different shapes and sizes. Those 3 balls, I have to pick 3. I'll pick those 3 which are representing my 10. That is what sampling is. So is going closer to the maximum value representing the whole data set? It is not. This is wrong. This is not exactly right. So later on what we did in another paper, we said maximum is not the ideal measure. You do the median of that. So median is where the values are upper and lower. So if you are closer to the median, means that you are representing the data set better. Is that okay? So that will take care of this problem as well. I have not fixed this in the slide. In another conference paper, we have proposed that instead of maximum, median is a better representation and it works better. So this is the kind of work which you do when you go to applicable AI. You take existing models and apply it to new data. That becomes data science as well, right? Machine learning applied to a certain data is data science. Is it making sense? Okay, so that's the first part. The more interesting part was spectral clustering, which I wanted you to focus rather than sampling, although I did tell you how to apply sampling to a data without telling you what the sampling algorithm is. Is that okay? Because we didn't cover it in the class. But spectral clustering, one of the most widely techniques, similar behavior, blah, blah, accurate, that's okay. These are the certain steps we do. This is a standard algorithm, 20 years old. I think 1998 or something, maybe 25 years old. This came out as a paper, and it has been used in various domains. And until 2020, the theory of that was still being developed. So it's important that people propose it, then they don't know why it works, and then you go about doing fundamental machine learning and try to tell the convergence of the algorithms. I mean, those are the people who are doing theoretical machine learning. So that is what we'll do. So what it does, it builds something called as a similarity matrix. What is a similarity matrix? If I've got 10 species, I'll find relationship of every species with everybody and make a matrix. 10 species here, 10 species here, build a matrix, similarity. Next, we find the Laplacian, which is D minus W. D is nothing but a diagonal matrix where you add all the columns, and W is your similarity matrix. You find its eigenvectors, and then you do k-means. So this is what algorithm does. Why it works is not what I'm going to tell you. What I want to tell you is how do we apply to this plant data. That is what I want to tell you. So that is why this is black, somebody else's work. Building the crucial similarity matrix. This is what was novel, which we did. So the very first thing we did was we built a vector of all the characteristic for every species. Now this, what is kappa? This extenuated X, this is the phenotypic characteristic 1, which could be the early plant vigor. The next could be number of P's per plots on NBB, SW, and so on and so forth. These are M of those are put in a vector format. I put a transpose because it's a tall skinny vector. Then what we do, we compute distance between two species with any of the measures. So these are the distance measures. Now the distance measure come into play, remember? We have seen few of them. It refers to many others. Beside Euclidean, square Euclidean city block is Manhattan. Cosine distance is the one which I've shown below. Hamming, you know. Correlation, Jacquard, you don't know. These are other distance metrics which are there. So we applied a lot of distance metric. What is a cosine distance matrix? It uses that cos of the angle between two vectors is that, and apply that and you get this, right? So now if you don't look at the last line, similarity matrix is capturing how two species are similar. Let's say I'm taking the first species here and the second species here, and this is my second row first column element. So this value should be large if the species are close to each other, similar, right? So which means if the distance between the two species is less, then this should be larger. Again, an inverse relationship. So 1 over d p1 p2. So we could use that. But when we experiment with it, we turned out that using, instead of 1 over d p1 p2, if you use 1 over e to the power d p1 p2 or e to the power minus d p1 p2, that gives a better spectrum of the similarity matrix. Why are we after the spectrum? Because eventually the eigenvectors will be used to do the clustering. So since it gave us a better spectrum, rather than 1 over d p1 p2, we did 1 over e to the power d p1 p2. Is that okay? Another decision when you're taking the existing algorithm, applying to a new data set. Okay. So now what happens? You took small n, a million species. You sampled them to 100,000 capital N species. You clustered the capital N species, 100,000. What will be the remaining 900,000 species? You'll have to cluster them as well, right? You'll have to map them to which cluster they belong. So we do something called as a reverse mapping. So goal is not just to cluster this small n, but the capital N. So the remaining n minus n species have to be put into clusters as well. So how do you do that? You find the distance of every non-clustered species with every cluster by adding that I am, for example, I am a new species and there are 10 clusters. I'll go to the first cluster, find the distance or similarity with all the elements in that cluster, average it out. With the next, average it out, next average. So where I'll find the minimum distance, I'll put it there. That is what this is doing. Is that okay? This is the distance which it's finding. Again, instead of 1 over dp1 pq, we are doing e to the power because of the earlier logic. And this is the normalization number of species in that particular cluster. If this cluster had 5, I found 5 distances and averaged the term divided by 5. This had 10, 10 distances divided by 10, correct? Okay, so overall we did this. So now the reason in the start of the class I asked you, tell me who is non-CS, who is not from computer science. No. You told me I want to do a complexity analysis. The CS people might be able to do it faster than you, but I want the non-CS people to help me with the complexity analysis. So this is what is worked out here. So I'll want you to tell me what is the complexity of each of the component. So let's say I was doing sampling. What will be the complexity of sampling? I told you that the complexity of running through is order n square, order n, sorry. That whether you choose or what you touch every unit once, there are n units, so it's order n. But how do you choose one unit is you compute the incursion probability. So what is the complexity of computing the incursion probability? That's what I want you to tell me. By looking at this, what's the complexity? Yes, non-CS. MN. Who answered? CS? Oh, you answered? Okay, good. Come. You also answered, blue shirt. You answered? No, you did. Somebody answered MN as well. You're mechanical, electrical? Oh, civil. Okay. What's your name? Why is it MN? Tell us, everybody.
Because there I is the one particular set and J is the one particular which goes to M and N. So we're doing 2 times as a matrix.
Correct, correct. So what he just said, if you look at this cost of this is M, because you're taking the maximum value and subtracting for all the and adding for all the phenotypic characteristics. So they are M of the phenotypic characteristics. So this complexity is M total. Yes? Yes or no, Shivam? Everybody okay? What is the complexity of finding the max? N, min n, complexity of this summation n. So shouldn't be order of n square? Look, you were checking every species 1 1 1, so that's order n is an outer loop. Inside now you're doing this computation. So my question is, this computation is taking m, so it's order m times n, which he said. But now I told you that the cost of max, min, max, this summation, all of these is order small n. So shouldn't it be order n square rather than order nm? Anybody can answer. CS can answer. Gaurav can answer. Yes, Yatharth. Correct. These are done once. These are run outside the loop. This is done only once. It's not done with every touching of the species. So max is found once, min is found once, irrespective of touching the every species. So that is done outside the loop. So that's why it's order mn. Is everybody okay? Comfortable? Should I move? Okay. Come, Yatharth. All right. So the next, so that is what we wrote. The complexity of the sampling is order m. What is the complexity of similarity matrix? This I'll tell you because I've not actually shown you the full algorithm. There is a cost. The cost of building the similarity matrix would be how much? How many species are you carrying? Capital N, the number of samples. N into n, n square elements. Each element requires how much computation? The girls? Each element of the similarity matrix will require how much work? This is each element is built. How much work it will be computationally? Yes, come. This should come like this. If you're CS, it shouldn't take just microsecond, it's M. because you're doing distance between two vectors P1 and P2 in a product which are both are m dimensions. You're just doing m multiplications and m additions. Correct? Yes or no? So the cost of building the similarity matrix is n square m. Laplacian matrix, I have not told you, there is an inverse involved, I have not shown you, so it takes n cube. Inverting a matrix takes n cube, take it from me. So that is what it is. Now somebody tell me why the cost of reverse mapping is n minus m times n times m. This is the last question. This is reverse mapping. It's as simple as this. Why is this cost n minus n times m? They are n minus n elements. You have to check with all the n elements which have been clustered. Now when you're checking it, the cost of computing e to the power minus dpq is m, because that's the distance you are again finding. Everybody with me? Gaurav, aajo. Nahi, come. I'm giving to everyone. I have to finish this. Okay. Take it. So that is the complexity. So the overall complexity is this, and complexity of hierarchical clustering is n cube. So we make it more than order of magnitude faster. Besides that, we are accurate. I think how much time do we have? We still have 5:30. There is a quiz. This room is booked for some quiz related thing. So I'll request them to take Kalidas, which I have already booked at 5:30. If they don't agree, then we'll move there. So then we'll see. So after that, what we did a lot of experiments. These were soybean species with 8 characteristics. We did some statistical comparison. We just didn't do it on soybean. We did it on maize. We did it on rice. It's independent of the plant which you're using. That's what we wanted to show you. We did some sampling analysis to tell how good our sampling is. Again, the focus of today's lecture is definitely not sampling, so I'm not telling you what these measures are. You can read from the paper. But what is more interesting is the clustering setup, because we didn't knew what is the ideal number of clusters. We went from 2 to n by 2, which I told in the class. We made these many clusters and we told which is the best clustering index, and that is what we clustered in eventually. We'll show you. To measure the quality of the results, what will we use? Silhouette values. This you know from the class. If you didn't know, you can see my recording. You have to do BI minus AI for every species divided by max of that. So we did 2 sets of experiments. So the first experiment is we compared our modified SC with pivotal with all competitive clustering with sampling, and then we compared with the basic HC. The sample size without loss of generality was 300, 500. This was, I mean, this could also be a hyperparameter, which we say, which you can tune in your model. So if you look at, this is the result. You see the similarity measure. This is the distance measures. The number of, I've skipped the slide where I'm showing you the number of, how did we choose the number of clusters. There's an eigenvalue gap technique. If you are interested and this relates to your work, then go back and look at the paper. There's a lot more work done in the paper, which was difficult to do in this much time. So what the theory said was, so this said basically where you stop, this was the eigenvalues we had computed of the similarity matrix, and you can see it's flattening out. So basically where it starts to flatten out, it says that is the ideal number of clusters you can pick. That's one heuristic. So that's why I picked 10, 20 and 30. Just a little left and right of 20 we went. So there are many other analysis done, like I said, in this, which I have skipped. We have done, this was sampling analysis and so on and so forth, but that's not what the focus is. So we'll come back to clustering. Go back to the table where we are in, and you tell me which is a better clustering and why. Okay, so this is the table. Number of clusters, this is pivotal sampling. VQ was another sampling I picked just for comparison, so you can ignore this table. This is our modified spectral clustering. This is our hierarchical clustering. And these are the Silhouette values which we get. So now if you look at these 2 columns, pivotal sampling with modified SC and pivotal sampling with hierarchical clustering, looks like it's, we are not working that well. correct? But then we went and analyzed it further, and we saw that the distribution of species between different groups in hierarchical clustering looked like this, and in R it looked like this. So now this one had a problem. Can somebody tell me what is the problem with this? Sorry? Yeah, so if you look at this more carefully, the Yatharth is on the correct lines. Most large number of smaller number of clusters have large number of species, and there are clusters which has absolutely one element. So if in a cluster there is one element, what will be its Silhouette index? Silhouette value. Somebody who's non-CS. I know you guys know it. Somebody behind. शिवम, अगर एक क्लस्टर में एक ही एलिमेंट है तो उसका सिलआउट वैल्यू क्या होगा? सेम होगा. कैसे? इट्स बी आई माइनस ए आई डिवाइडेड बाय मैक्स ऑफ बी आई ए आई. वॉट विल बी ए आई? Let Shivam answer. Yeah. What will be AI if I am the only person in this cluster? What will be my AI? 0. So it is BI over BI. BI over BI is 1. Silhouette value 1 means this is an excellent clustering. It's misleading. I am the only one in the cluster. My Silhouette value is 1. So it is artificially inflating your average Silhouette value. So that is what we have put stars here. That all these values, although they look higher, they're artificially inflated by one element in these clusters. So again, why Silhouette value is not the Bible of telling whether a clustering is good or not. There are many other measures which you use. That's why together with it. Hai? So this is one of the flaws. So then we realized, no, the biologist said your clustering is better because you're seeing a distribution. This is the distribution we want. Hai? So then we did it for 300 and we got this result, which you can see we are getting better than VQ. Then if you look at our final result for k equal to 10, we compared our algorithm with the hierarchical clustering. Our algorithm is spectral clustering with pivotal sampling. HC is hierarchical clustering without any sampling. This is the algorithm which the plant biologists are using in their software. So you realize that for this distance, we gain up to 45% in the accuracy. Our Silhouette value becomes dramatically higher. And in this one also we get dramatically high. You can see that for all distances it doesn't work that well. So for a particular set of distances we are really good. For some we are good and slightly bad as well. But that is not what we are after. We are after the best value. So we looked at this. So the summary of this is we are up to 46% more accurate than the existing technique. But we are also in magnitude faster because we have done sampling. So if you look at the complexity of the algorithm in terms of the floating point operations, if n was 2000, m was 8 and the sample size was 500, you get this number, and otherwise you get 10 to the 10. So you are like 100 times faster. In this one you are 1000 times faster because of sampling. And the interesting part is because of sampling you're not losing, in fact you're gaining, because the algorithm is more accurate. Spectral clustering itself is accurate. Even that you're working on a smaller data set is giving you good results. Is that okay? Everybody good? You got a flavor of what I'm trying to tell you? Yeah. So this is the paper which I just showed is uploaded. This is the paper on genetic. This is some work on pivotal. Achha, 2002 was where spectral clustering was 20 years ago informed, neuro IPS. So yeah. Is that okay? 5:22. So we have still 5-10 minutes. Yeah. Yeah, please. Hmm. Correct. Well, the thing is, we don't only do it once, you have to repeat it with the new species getting evolved. That's the problem. That this, now that you've gone through the corona phase, every time a new mutation comes, its sequencing is done. Then you get a new sequence. Now you have to cluster that as well. So new species keep coming. You're right. If we had a fixed set of species, and we had to do it only once, then probably we'll want to do it as accurate as possible and not do sampling. But because new species keep coming, we have to want to do it sampled way and do it faster, basically. Is that okay? Yeah. Okay. Come. Did you get it today? Hey, you're working on rice, na? Are you doing clustering of rice? No. K-means is very simple. And let's go through that clustering PDF. I've used that many times, the table specially, to tell me which one I should pick. Yeah. Okay. So before we start, I wanted to just discuss few things for the class before we close. And then 5:30 we'll start. You got your laptops, those who want to discuss the survey? है ना? ठीक है, okay. So this was about some comments which I got along the way about the feedback of the class. ये क्या हो रहा है? The color is fancy. आ रहा है ना ये? हाँ. So few things which I have addressed and few things which can't be addressed. I just wanted to tell you because that still half of the semester is left. है ना? So what you liked about the class was the pace was, I think some people said it's little fast, some said slow, but mostly it was okay. Explanation, the way I explain is something which you liked. So that is what is, and then the organization, है ना? Which is most, and one thing I wanted to tell here, let me write it. Organization was fine, including the Google Drive and as well as the YouTube. So the why did I upload it and why do I take so much pains of uploading it on YouTube is so that you can go and revise. And if you have the certain concept you have forgotten, it is not for Gaurav. So it is not for students who don't show up in the class and want to study. That is not what I didn't make the recording so that you sit in the hostel and not come to class. That was not the goal. The goal was that you go back and revise and what you've forgotten. When I took courses, I wish that there was a recording facility, but that time it was not there. Coming to so this was fine, and coming to the improvements which few which I have done and few which cannot be done, I wanted to tell. One was the practice problem. So if you have seen now that we have covered 3 models, we did questions in class. There was some in tutorial, but there are a lot in practice problems. And you have not even done all of the practice problems, which I came to know on Tuesday. When this guy did the questions, the 20 odd students over there, they had not done all the questions, but you should please do it. The second question was more on the real life aspect. So this, like I said, I didn't have time in the class to cover. So I assumed that your survey paper will do it, hai na? Which I think is helping. Did it help? You got a better perspective of real life, hai na? And the third thing was that some people wanted coding. This is there in the lab, but then I came for in the labs deliberately. I usually avoid and let the TAs do it, but I came to all of them so that these guys are also more serious. And then if you have any doubts, so those things I covered. The 3 things which is not mostly in my control, and one was a lot of people said math was a little overwhelming, but this feedback I had taken like in the second lecture of SVM, and we had not even finished SVM, but I assumed that now it will be okay. Is everybody okay with the math? But if it is not, you can ask me now also. Make sure you understand the matter. Remember, machine learning is not about using, ये बंद कर देना, machine learning is not about using Python libraries. That is not machine learning which you're learning. It is about knowing the models, and in future maybe you go and work for Google and you find a new model. Why not? You can always do that. You make a model for your application. So this math part, unfortunately, I couldn't make it more simpler than I've done. There was a recording for you to go over it, but if you're still confused, you can ask me now. Ask me tomorrow. Anytime you can ask before the exams or after the exam as well. Then there was a concern about rest of the semester how this is going to be. So look, Professor Chandresh is a different person. He teaches differently than I do. So what you could do is request to him that if he can create recordings and upload. है ना? Other people have other ideas of recording. Like I said, if I record, then people don't show up, which is happening here. Na? So if he agrees, he'll record. If he doesn't agree, he'll not record. He has his own style of teaching, which I can't change. I'm not his boss. Hai na? So I'm a colleague to him. So you can think of it that way. Although I'm overall coordinator for this, but he has his own style, which I'm sure you would find it, you'll like it. It may be different. The last thing, there was one more thing which somebody asked, and I thought math was there, rest of the semester was there, which I couldn't actually cover. Choosing the model. Choosing the model or more on the intuition. This was something which, so these are the three which I have implicitly taken care of. I can't do much about them. These are all done. So look, choosing a model is something which will happen more with practice. Like even when my student, he's MTech, he's doing a relationship between phenotypic data and genetic data. Let's say you've got this plant height. Which part of the sequence in this ATGC is telling you that my plant height will be 2 meters or 2 centimeters, or which one is telling you to figure that out? So this correlation, a lot of these plant biologists do it by statistical method, which is not very accurate, takes a lot of time. So you're trying to use machine learning. When we go about using machine learning, still I have, with so much of work experience, I have no idea that this is the ideal model for it. So we applied random, so trees you would learn towards the last topic in the class, random forest we use, boost we used, ADA boost we use, support vector regression using support vector machine for a regression task is a support vector regression. So then we realize support vector regression is working the best. Now we are trying to figure out why is it working better than the other models. So that's the biggest problem is machine learning, that the intuition of why a thing works or does not work comes after you have tried it rather than at the start. So it's very difficult to tell which models to try. But in general, if you're doing a classification task, if it's an imaging task, you will say use a convolution neural networks. If it's a temporal task with a sequence of data, I'll say use a recurrent neural network or use a LSTM, or I'll say compare it definitely with the SVM with the advanced kernel, with the non-linear kernel, if the data is highly non-linear. So those are certain thumb rules we would know. If it is a regression task, I'll say, okay, use the basic non-linear regression with regularization, because you know you would use. So there's certain set of models which you would figure out to use, and then you have to figure out why this model is working well. So there's no direct answer for this. Hai na? So all these things don't have a direct answer with me. This will come with you more with practice. Is that okay? So we'll stop now. This is recorded also. Hai na? Those who don't come can see this. And then we'll now talk about 5:30 is already there. Yeah, okay, so we'll see if people are waiting for. Stop the recording. Hai na?